Welcome to Part 4 of Adenovirus and North American Cervids. During this session, members of the Wildlife Health Working Group from the Western Association of Fish and Wildlife Agencies share their experience with adenovirus in various wild cervid species. Dr. Todd Shurey, a wildlife veterinarian with Parks Canada, will share his experience with adenovirus in wild mule deer in Alberta.
Just talk quickly about an outbreak we had in Waterton Lakes National Park in 2006. Can you see my screen?
Yes.
Okay, great. Presentation mode here. So Helen referred to this. So we got notification from some of our staff in Waterton Lakes National Park in the summer of 2006. So it's located way down in the southwest corner of Alberta. So it borders actually Montana and British Columbia. It's a small little national park, just north of Glacier National Park, right on the edge of the mountains, the foothills. And so the town site is pretty small, sits right on the edge of a lake. And, like a lot of these mountain towns, there's a herd of mule deer that lives in the town site pretty much year round. Because a lot of the town site residents like to feed deer during the winter and all year long. And so there's typically about 30 to 40 mule deer within the town site that pretty much stay there all year round. And they drop their calves pretty much right in the middle of the town in May, typically early June, cause a lot of issues with visitors who flood the town site in July and August. So we got first wind of this in, I think, July sometime, but it took till August till we were able to get a couple of the fawns to a local practitioner, veterinarian in Pincher Creek to do a necropsy on a couple of them. So that's kind of the story and I'll tell you a little bit about it as we go here. So there were a total of 13 fawns that we found dead within the Waterton Lakes town site between roughly about July 4th and October 19th in 2006. And as you can see, these fawns are typically born in the town site on people's front lawns or in green spaces. We had two of them necropsied in August, and they had very grossly visible pulmonary edema, thoracic fluid, and large lymph nodes, mostly bronchial and mesenteric. Some of these fawns were observed in respiratory distress before they died as well. So coughing, terminal convulsions, but they usually died fairly rapidly. And there was some rectal bleeding in some of them. And on histopathology, pretty much what has been described previously by everybody else. There's a generalized vasculopathy characterized by endothelial swelling and very obvious inclusion bodies. So I got actually Dr. Gary Wobiser at the WCVM to help me out with this. And he did some immunohistochemistry with a bovine adenovirus 5, which stained positive for this, and also sent tissues to Leslie Woods. And she used some immunohistochemistry for AHD specific from the California outbreak. So I think at the same time, we've also alluded to this a couple of times. Helen's staff had captured some elk in southwestern British Columbia, just across the Continental Divide. It was 34 elk, I think, that they captured in the winter of 2007, so roughly a year later. We did some serum neutralization on that, and 79% of those elk were positive. I think the cutoff we used there was 1:32 titer. So that was quite interesting. So obviously there was, some more exposure going on in the in the larger regional area. But I think just because this was in a national park and a town site, it was much more noticeable. So there's likely other stuff going on in the wider general area. If you just look at the kind of the epidemic curve for this, there was a couple fawns in middle of July, and then a couple at the end of July, and it sort of peaked roughly end of July, beginning of August, and then there was a long sort of tail out period. We had a couple in September and October, and they were all fawns of the year with the exception of one that was a yearling, and they're all mule deer. And there is a herd of elk that lives near the town site, not in the town site as well in Waterton Lakes. And there was no mortality observed with any of the elk as far as the staff could see. So just a little outbreak there, and we'll see if that adds to the discussion. I'm not sure it answers any of the questions that Leslie posed. There's no sort of obvious nutritional stress that we know of going on that year, or environmental conditions that were sort of different as far as we know. So I'll stop sharing.
Todd, have there been additional cases show up since then in wild animals?
No, that was the only year we saw mule deer fawn deaths. There were a couple of years where we saw the odd one, but nothing like we saw in 2006. So.
Okay. Next up, Dr. Kimberly Beckman from the Alaska Department of Fish and Game shares her experience with adenovirus hemorrhagic disease in moose and surveillance in cervids in Alaska.
All right, so I'll just quickly go through what we know in Alaska. I didn't mean to jump here, and this comes from work that I did with Kathy Burrick-Huntington and Myrna Miller. Previous to the cases I'm going to describe, there was some serology that was done by Randy Zarnke and Howard Lemkuhl, and notably, they did have some seropositive caribou. I think it was Helen who mentioned we didn't have cases in caribou. We have some evidence of exposure, at least with antibodies, but this was all serum that was frozen from live capture released animals. There were no reported mortalities that anybody detected. So then what happened where I got involved in this was there were two calves that were orphans that were at the Alaska Wildlife Conservation Center that died within 2 weeks of each other during a really severe winter conditions. These animals are highly stressed. Their nutritional condition and things at this facility are not very conducive for long lives in these moose. So I would say they did have a severe stressor happen to them when they died within 2 weeks of each other, and they had the same kind of symptoms, clinical signs that everybody else has been describing. And the lesions were the same in these moose. Has been reported pulmonary edema, hemorrhages, enlarged lymph nodes, and palatine ulcers. The second case again had the pulmonary edema, and this what turns out to be a thromboembolus in the lung. Lots of hemorrhages in the intestinal tract, especially the cecum. There was ulcers in the mouth and ecchymotic hemorrhages in the kidney. Goes along with the idea that there's some DIC or something going on. Histopathology, the same as everyone else has described in the deer and elk. The endothelial cells with inclusion bodies, hypertrophied endothelial cells, lots of edema. The one unusual finding that no one else has been describing are these syncytial cells that had inclusion bodies that were found in the epicardial fat and the cecal serosa. Remember I said there was a lot of hemorrhage in the cecum in the one animal, and also found in the lamina propria. So that was the unusual finding. Aerobic culture, one of the animals had the usual actors for secondary infection, but PCR was positive on nasal swab and lung tissue from both animals. And we also isolated this mycoplasma that seems to be quite prevalent in moose in Alaska, and the draft genome of this is actually in press right now. It's a new species being described. The electron microscopy of endothelial cells from the heart was positive for virons. Kyla Kaufman from University of Wyoming was able to genomic sequencing on this and identified it as the genotype B. And this is a slide from her that shows that Alaska and Nevada and some of these western states all predominated with that type B, but that was quite different from what is common in the West Coast of the U.S. So at this facility, these calves were in a pen with some adult moose. Like I said, they underwent a severe stressor. There's also elk, reindeer, black-tailed deer, bison, and muskox at this facility. The only new introductions were 2 muskox calves and a deer fawn. But they weren't housed really close to the deer fawns, but they were right next to some elk. We tried to get nasal swabs from these other animals, but we didn't get very many samples. So I can't really say it isn't there, but those were negative. We looked then at a bunch of nasal swabs and tonsils and retropharyngeal lymph nodes from free-ranging animals and didn't detect anything until we had this one deer from Southeast Alaska that was hit by a vehicle and euthanized at a vet facility. And that one, it was detected on the tonsil. But again, we looked at a whole lot of tonsils and nasal swabs and didn't detect it anywhere. So these cases are going to be published in the next issue of AHD, and there's also, in JWD, and Kyla Kaufman also has a paper in there on her recent study on adenovirus. So that's
okay.
it.
Thank you for that, Kimberly. And do you have any speculation about how it gets around?
I really think that it's there's a latent carrier at that facility, and I suspect that and kind of what Leslie was saying too, that's got to be I think there are carrier animals that we just can't detect. The antibodies are not either not protective or just wane really quickly. And then when there's a stressor, I think that's what causes outbreaks. I think that with this particular case, it really seemed like it was, some kind of a stressor because the weather was really bad and these animals were highly stressed and they died within 2 weeks of each other. And there hadn't been any recent introductions of anything. It had been over a year from anything else being introduced in this facility. So it has to be that there's a carrier there.
And I think, Karen, you have a question right now. Where are we on the ability of serology testing? Does someone have an answer to that? Just unmute yourself. And
the state of Oregon is developing some serological work, and I think that's available, but maybe somebody from Oregon can.
This is Nate from Nevada. That's where we've sent all of our serology.
Okay, so the serology is happening in Oregon from the sounds of things.
Yeah, I wanted to ask if Colin was on, at the WDA a couple of years ago, I had asked where that was. I was really excited. It's an ELISA, and wanted to compare our serum virus neutralization results with the ELISA results. And, at the time, Rob Bildfeld said that he didn't think it was ready for prime time, but I wanted to check with Colin to find out, is it ready?
The last that I heard about this was that it was near ready. They had not pushed it to any level of being, I don't know, even close to a validated state, of course, but they're still working with it. Bring Rob back if you would. We miss him.
Can everybody hear me okay?
Yes.
Good. Hello, everybody. There's many familiar faces, as Leslie said. Leslie, it's been a very long time since I've seen you. It's nice to see you again. We put together, actually, Jessica put together this map of just the number of adenovirus cases that we've seen in the state over the last 10 years. And it's amazing, as many times as Wyoming is mentioned in this discussion, you think we'd have a lot more adenovirus, but in reality, we don't. Of course, we have Dr. Miller, and then we had Dr. Woods, who did a lot of work on adenovirus. So just briefly, in the last 10 years, we did see a a significant increase in the number of cases in 2015, 2016. We saw 18 cases. And then pretty much over the last several years, we just haven't seen very many cases until this year. I think we've documented 3 cases in 2020. So certainly there's more adenovirus out there than we're detecting. But for the most part, at least in Wyoming, we just don't see very many cases. And of course, most of our cases are in whitetail and mule deer. Only 1 case in pronghorn, and then of course the moose that was mentioned earlier. So really, it's not a big disease concern, except in a few locations. Of course, you can see in that lower southwestern corner of the state. That was in conjunction with a mule deer research project, and obviously they did identify a few cases of adenovirus, and that was in fawns.
I'm going to ask a question here since we have one in the chat, and it's really about outbreak control. So for any of the outbreaks that have been described, are there any measures implemented to control and decrease risks of transmission between cervid farms or within wildlife? Anyone want to hop in there? And
yeah, I think I talked a little bit about case management and the messaging that we provided to folks, hunters and residents of our particular area. I also wanted to comment that in terms of transmission in our case, we know very well that deer swim between these islands and they can swim for very long distances. So the local transmission is fairly easy to explain. However, we have zero idea of how it got to the original islands. They are remote from the main island. They're remote from the mainland. They're connected by ferries. So I guess it's possible a hunter had come back with something that was contaminated, but it's a real head scratcher. No real idea.
I want to just call on Anne if you wanted to have any comments about your experience in Arizona, if you're able to, if you're still on the line.
Yes, I am still on the line. I'm still on the line.
Okay.
So what we had happen here in Arizona was we have a zoo in northern Arizona that had 3 cases identified in 2018 that were determined to be PCR positive through Leslie's lab at UC Davis. They were submitted by the local veterinary school, Midwestern, and the pathologist there saw, I think, some of the typical signs. The one thing that was a little unusual about these cases is they were 1 to 3 years old, so they weren't fawns. At the same time in that year, we had an outbreak of EHD in several locations, including up in northern Arizona. So next time I had the opportunity to do some serial surveillance was just this past year. And so we tested some of the deer and elk that were in the kind of the same vicinity. And interestingly enough, we found that we had really high seroprevalence in those populations. And the testing was done by Oregon. I believe they did a serum neutralization test. So we had about 19 of 26 mule deer test positive and 15 of 15 elk test positive. Some of the titers were just kind of in the moderate range, but we had titers up to 1,024. So that was really interesting. We've never in wild cervids detected this disease. I'm going to be looking for it for a lot more carefully. I'm concerned with the AHD blue tongue outbreak, whether or not you can have comorbidities, so dual infections. I had a couple of EHD cases that I thought had really, really extreme pulmonary edema on gross, and I think I've got tissue saved back, and I'm going to try and get those tested for cervid adenovirus as well. And that's basically where we stand here in Arizona at the moment.
Okay. And any hypothesis from your end about Helen's questions about how did it get to
So
to her.
interestingly enough, I'm also dealing with an adenovirus problem in mass bobwhite quail. And we've been following it in this captive breeding facility for about 3 years now. I really do believe that there are inapparent carriers. We have had no luck detecting the virus in between outbreaks using cloacal swabs or oropharyngeal swabs. I think the only way to find it is going to be doing tissue testing of some sort, which the results with the lymph nodes suggest that. So I do believe there's probably inapparent carriers with these adenoviruses. You know, the sporadic nature of the disease and also the co-occurrence at least in Arizona here with the outbreak on the EHD events. I know adenoviruses aren't supposed to be transmitted by insects, but never say never in my book. So I would not discount that 100%, but I do think the inapparent carriers is probably a very high likelihood with a stressor on top of that, maybe another infection that we're not detecting. Who knows?
And Kristen, did you want to share your experience? You had said you had subclinical AHD detections.
Yeah, I just reports. We did have an outbreak in July of 2017 in mule deer fawns in Goldendale, Washington, which is about 140 kilometers east of Portland along the Columbia River. It was a small outbreak. We estimate only 70 deer died. The diagnostics were not super clear cut. Grossly, it looked like classic systemic cervid adenovirus. Samples were submitted to Waddle. One submission was too autolyzed for good histology, so they couldn't identify inclusion bodies, but a sample was PCR positive from that submission. And the second submission did have suspected inclusion bodies in the endothelial cells, but it was PCR negative. And I'm sorry, it did not state which actual samples were tested for PCR. So not a super clear-cut diagnostic here, but concurrently, when we experienced this outbreak across the river in about 20 miles to the east in the Dalles, Oregon, they were having a large adenovirus outbreak at the same time. And so that's an interesting situation to me. As Helen stated, deer do swim pretty well, and we do have photos of actually elk swimming the Columbia River, so perhaps that's how it jumped over into Washington. But that is our first and only case to date of an adenovirus outbreak in mule deer or any deer in Washington. The one possible subclinical situation that you alluded to, this was just, we were collecting young elk in the south central part of the state to serve as negative controls for our hoof disease investigations. And as part of that investigation, we requested virus isolation. And from 2 healthy young elk, these would have been 7-month-old elk, adenoviruses were isolated from tissue pools. But this was done at the University of Wyoming. However, the more specific type of adenovirus was not reported. So I don't know if it could have been a subclinical bovine adenovirus or what, but these were subclinical elk. So I don't know if that really adds much to the picture. So other than that, it's just kind of almost our anecdote from Washington.
I am going to just ask Nate if you, from Nevada Department of Wildlife, I'll get it right this time. Do you have anything you want to jump in here with your experience if you're still on the line?
Yeah, I'm still here. So I guess I'll put the caveat here that most of what I'm talking about is based on the serology, and it's mostly from Oregon. So it is, as Colin said, not validated. And yes, Colin, we missed Rob too. So the couple of things that we had was we did have a couple PCR positives out of eastern Nevada quite a while ago. I think in deer, those are mule deer. One of the most interesting things in deer is there was a die-off that anecdotally had symptoms or pathology similar to adenovirus in the Toyabe Range in central Nevada sometime around 2014. And when we did captures there in 2018, we actually got 81% of the deer seropositive for adenovirus. So it's a small sample size. I think it was only about 15 deer that were done. But it does have, you know, maybe a little bit, if that serology is accurate, maybe a little against the kind of that serology is uncommon. And then the northeast of Nevada, there was a herd in area 7 that had about 10% seropositivity. In elk, we had done some various surveillance and hadn't gotten any positives until this year. We have a mystery kind of elk disease that's characterized by pericardial fusion and some non-suppurative encephalitis, and those, at the suggestion of Dr. Taylor at Waddle, those were tested for adenovirus on serology. We had 6 animals from that area, or 5 animals, and all were positive on serology. 2 of them with very high titers at the maximum range, which is greater than 1 out of 4,000, 4096. And we did test 2 of them by PCR on lung, and all had virus isolation, and those were all negative. But given some of the talk of having difficulty finding it, maybe in these chronic, maybe these are more chronic ones, I'm kind of interested if there's other tissues or other methodologies that people might suggest we continue to follow up these elk on. And because of those positives, we're doing some broader serologic surveys this winter during our capture season, both in mule deer and elk, especially in that northeastern part of the state. So that's all I have. Yeah, I'm curious on the testing of, too, if anybody has suggestions on that.
Anybody want to jump in on that? Suggestions on testing for Nate? Okay, if you think of anything, put it in the chat, and I'll just ask if Karen Fox from Colorado, are you still on the line? Okay, so Karen says her audio isn't functioning, so she's listening on the phone, but she'll type up some notes here about Colorado while we wrap up. So I think what I want to do right now is just, I'm going to stop sharing my screen, and let's just take 10 minutes and open up the discussion to, well, what do we think? Jump in. Helen, if you have specific questions that you in particular want to pose of anybody, now is a good opportunity.
Really, the discussion has answered my issues more than anything else ever could have. Just simply that we don't know enough about it, and we may have missed this disease in the past, and that may have served as the source.
Okay. Okay, and Karen's comment here on Colorado is we have sporadic cases in young and immune-compromised deer, rare cases in elk, possibly pronghorn, although those are usually based on PCR without confirmatory inclusions in endothelial samples. Okay, and then Anne is answering Nate's question. You might check the lab results for the method. So there was a question about testing. The ELISA hasn't been validated, but they also run serum neutralization. And that's what was run on her samples.
So I have a thought to throw out there. I think that mule deer, especially black-tailed deer, are exquisitely sensitive to the adenovirus. We did ad nauseum testing to rule out everything else, and those deer during the outbreak, and all of our outbreaks in California, With the systemic infection, these have been animals in very good body condition and didn't seem to have any stressors. And based on the experimental, I think they're just very sensitive. But we did have some deer that did have antibody response, 12 to 15% down south, and we never saw any mortalities. So I think that maybe those deer are less susceptible. And I think white-tailed deer are less susceptible. And based on what I've heard about the elk, that maybe these are more immunosuppressed cases. You know, correct me if I'm wrong, but then we start seeing the antibody seroprevalence in those species. So if that's true, then maybe, you know, maybe like samples should be collected from the less sensitive species, like in a region when you have an outbreak for a diagnosis, and you will see an antibody response in species that are less susceptible. So I'm just kind of throwing that out there for discussion.
Okay. Want to jump in there? Feel free to unmute yourself and jump in.
This is Colin. I have something related to that, Leslie. We have seen in 2000. I'm trying to think of the years now. I don't have it in front of me. It was around 2002 through 2004. If you recall, we did a lot of serology and we tested everything. Jack Mortensen was here helping with that, and we ended up getting serology hits in elk, as well as whitetail, mule deer, blacktail. We had a lot of serological hits in all those species, subspecies, and at that time, you know, it seemed to we seem to have a lot year after year. But now, and I'm curious if this happens in California, we see it every year, and we get to where we have probably some surveillance fatigue, even amongst us, at looking at this. But where we notice it, of course, when we fill out those squidus hemorrhagic disease reports, every year we're putting down some adenovirus cases, specifically in places like Bend, usually in fed herds, you know, by the public. And we, in fact, I don't even think we do any deer fawn rehab anymore because we had so many outbreaks in the deer fawn rehabbers. I mean, they would lose 20 at a time, and which I think you saw in California also. So I'm just curious, is California seeing kind of a constant background of this popping up?
Yes, in the retrospective study, we had a little flurry of activity with 6 or 7 dying from a herd, and then it stopped. So we diagnosed adenovirus hemorrhagic disease every year except for 3 since the big outbreak.
Okay.
And so we have little flurries and then nothing, and then only 3 years without, and then you have a huge outbreak.
Yeah, I think that's what Julie and I are seeing too, at just a different level.
Yeah.
Okay, there's another question in the chat here from Karen, who's wondering about missing cases of chronic presentation within deer and elk, sometimes given a presumptive diagnosis of chronic wasting disease. So I'll just open that up to everybody if you have some thoughts about that. Are cases being missed because they have a presumptive diagnosis of CWD? Go ahead and unmute yourself if you have an answer.
I mean, I think that the severe necrotizing, if the necropsy is done, the severe necrotizing lesions in the upper alimentary tract would tell you that this is adenovirus. But if a necropsy isn't done, then yeah, they kind of present similar. They're wasting and they drool. So
okay. Okay, and Helen says not in BC. Understand. So, and then Margot's saying not in Alberta, we test for presumptive CWD. There's a question here too from Anne about what about the possibility of strain differences in pathogenicity?
Well, I kind of threw that out there when Myrna did the sequencing, and the one strain that I sent her was a 9394 isolate, and she did find a deletion in that variable non-coding region that was different from other deer. So, I mean, like I said, it's an N of 1, but it's really interesting and should be pursued.
Okay. All right. Any other questions or comments that anyone wants to raise while we have such an incredible powerhouse of wildlife health specialists on the line? And I just want to say a huge thank you to all of you for enabling this and for being willing to share your expertise. I think this has been a really great opportunity, both for our community for emerging zoonotic diseases, as well as for wildlife health. I know for Helen, I hope that this has helped answer some of your questions, and for Jennifer too, having seen that first case in farmed elk in Alberta. I would just say a really big thank you to everybody, and hope we can do something similar again sometime. So thanks all, and I will stop our recording and say thank you, and have a lovely afternoon the rest of your day.